Hello, Abu Dhabi University and everyone. I would like to say it's an honor for us to be presenting for the 9th undergraduate research competition. My name is Yamama Al-Namer, and here is my colleague Rima Tahrawi. Today we will discuss our literature review on the clinical outcomes of COVID-19 in recipients of T-cell therapy and strategies to overcome challenges encountered in that context. Basically, we've divided our presentation into 4 parts. We'll start with our introduction, followed by the methodology. Then we will be discussing the results, ending up with the conclusion and future directions. Let's start by having an overview on our topic. So our new coronavirus strain named as the severe acute respiratory syndrome coronavirus 2 has been named by the WHO as the 6th top public health emergency of international concern in 2020. In the majority of patients having the severe form of the infections, high levels and expressions of pro-inflammatory cytokines have been witnessed, including the interleukins of different types, the interleukin 268, 268, 17, and B, as well as the granulocyte growth factors, macrophage inflammatory protein, tumor necrosis factor alpha, and the interleukin gamma inducible proteins. Also, in these patients, this is what we call the cytokine syndrome that is related to the cytokine release syndrome faced by patients having the cellular therapy treated for their cancer, especially the lymphoma and leukemia type. For the chimeric antigen receptor T-cell therapy, these therapies target specific patients, and when they are administered to patients with COVID-19, they end up with the cytokine storm that can cause target organ damage, toxicity, as well as local and systematic tissue damage, and also they can cause unregulated innate and adaptive immune responses. When they get infused into the patient's blood, as you can see in this slide, in the picture in the next slide, they proliferate and then they get the capability of killing the tumor cells, releasing their antigens that are taken by the antigen-presenting cells to be recognized by the cytotoxic T lymphocyte, resulting in eradication of the tumor and preventing its reoccurrence. However, it is important to study the favorable and unfavorable outcomes of using such a therapy in the context of COVID-19, and also to study how it affects the vaccine administered to these patients, and to develop strategies to overcome the challenges faced. So we aim to review literature on that topic to make sure that each patient's need is met. We did so by searching into specific databases like the Google, PubMed, Embase, and Elsevier using specific keywords. So we ended up having 50 articles because of the novelty of the infections. So we narrowed our search to include articles written in English and those that are research-based and published in healthcare journals. At the end, we selected 21 articles. Let's start by starting talking about the first theme, which is favorable outcomes of COVID-19 in recipients of T-cell therapy. It has been studied by many researchers, and many of them stated that the overall survival was very high in this population, being 78%. And in another study, it showed that using this technology, using this idea and developing a vaccine resulted in the recognition of the T cells of the C6 of the receptor binding protein of the vaccine, resulting in the phosphorylation of the S6 protein, which ended up having high expressions of a lot of cytotoxic agents like the perforin, granzyme, Fas ligand, and the interleukin gamma. And this can mount a robust immunity against the infection. Similarly, in another study, these patients had high expression of the CD8+, CD4+ cells, CD4+, PD1, as well as many other naive type of these cells, which are also cytotoxic and can help fight the infection. Also, in another study, administering the T cells to such patients resulted in a success in the therapy, moderate cytokine release syndrome, but was well tolerated and finally negatively affected COVID-19 in such a population. Moving on to the second theme, which is the unfavorable outcomes, many researchers have found that patients express high levels of inflammatory cytokines like the ones aforementioned, and they are related to the cytokine release syndrome. And according to Mashtag and his colleagues, these patients had abnormal chest x-rays where 28% of them had hypoxia and 19% of them went to the ICU, where 10% received mechanical ventilation. The most important cytokine here is the interleukin 6. So an inhibitor has been developed, which is tocilizumab, and the researchers agreed that by administering this type of agents in patients in a medium of 2 days followed by mechanical ventilation, will end up having less secretion of the interleukin 6 and tumor necrosis factor, thus less ICU admissions. Also, patients had a lot of complications like the GIT hemorrhage and the hepatic, renal, cardiac, and respiratory dysfunctions, and they also experienced expectoration, fever, and cough. This brings us to the end of my part. Now my colleague Reem will continue on talking about another theme. Thank you.
So, hello everybody, my name is Reem Al-Tahrawi. Now inshallah I will complete what my colleague Yamama started. So moving now to the third part of our presentation, which is the results, which is the strategies to overcome the challenges of T-cell therapy administration in context of COVID-19. So based on the impacts that my colleague Yamama mentioned, it was necessary to have new strategies to conduct them in order to avoid these impacts. So many researches were conducted to support these strategies. The most important strategies are like, first of all, we have to establish a triage algorithm to delay the use of CAR T-cell therapy as much as possible we can and prioritize the patients. Also, we have to ensure that we have an enough number of the cellular treatment team that is always available once they are needed to respond to COVID-19 concerns. Moreover, it's important to apply the protective measurements and the protective equipments for both the staff and the patients, and having the personal protective equipment, the PPEs for the staff, in order to reduce the transmission of infection from the staff to the patients as well. Moreover, we have to conduct regular physical monitoring, thus leading to monitoring the patient in a regular way. So we discuss his health status in a regular way. Moreover, we can support the virtual rounding team as well as using electronic communication devices for the patient visits, which can reduce the patient visits, thus reducing the transmission of infection from outpatients. And for the last part of the results, which is vaccine-induced antibodies and responses of T cells. Due to COVID-19 pandemic, vaccines have been regarded as a key strategy to prevent the occurrence, morbidity, and mortality of the infection. Fortunately, we have two messenger of mRNA-based vaccines. We have BNT162b2 as well as mRNA-1273. Both are recently available for COVID-19 vaccines. They have demonstrated their potentiality for inducing the protective immunity against the virus in the community. There was one cohort study. Its finding indicated that the level of RBD IgG was massively lower in the CAR T cohort in CAR T patients. Also, one study that was done by Oakland and his colleagues conducted an analysis on SARS-CoV-2 spike binding IgG antibody levels in multiple myeloma patients. The findings showed that we have 84.2% of the patients that are fully immunized. They have shown measurable SARS-CoV-2 spike binding IgG antibody levels. Also, we have one study that was conducted in 2021, which demonstrated that 60% of the patients treated positive for COVID-19 antibodies after the vaccination. While we have another cohort study showed that we have 90% of the patients with cancer and 100% of the controls were found to be seropositive following the administration of the second messenger mRNA BNT162b2 vaccine dose. Similarly, we have one other study that found out that we have, after post-vaccination, naive patients where 65% of them and 29.9% of them developed IgG and IgM, while on the other hand, we have 35% of them showed SELI spot positivity. Moreover, one prospective cohort reported that we have 94% of the participants achieved seroconversion. It's assumed that the reason behind the findings that CAR T cells might result in antibody and T-cell responses to the vaccines, leading to the depletion of beta cells. This brings us to the conclusion and the future direction of our research. COVID-19 impacts have extended, affecting cancer patients receiving cellular therapy in terms of morbidity and mortality. Therefore, actions must be taken to provide prophylaxis in this immunocompromised population. Moreover, due to the gap in the literature in this context, research must be conducted to bridge it. Future studies should be done on the efficacy and the safety of alternative cancer treatments like anti-angiogenic therapy in this context. Also, further research must assess the efficacy and the safety of using pre-exposure prophylactic antiviral monoclonal antibodies in this population. Finally, rather than relying only on in vitro findings, it's better to have some clinical studies in human subjects on developing an alternative strategy to vaccines target COVID-19 by involving CAR-Ts should hold place in order to get more reliable and applicable results. Here you can find all of the references that we have used them in our research. Thank you so much for watching our video.
Thank you.